This afternoon, we're still in the library of the Beckman Center at Stanford University. This time we're talking to the first of two groups of engineers. These are the people who actually built the cell sorters that we heard so much about, and we want to find out a little bit more about the history of the cell sorters from the engineering and technological points of view. The people we'll be talking with, right to left, are Richard Sweet, Nicholas Veizades, and Richard Stovel. Maybe we should start with you, Dick Sweet, because part of the story starts even before Stanford or anybody else even has begun to think about cell sorters, and namely that's your work that you did on inkjet printers. Could you tell us a little bit about that work?
This is a project that I worked on in the Applied Electronics Laboratory here at Stanford, starting in 1961. And the goal of this work was to write with small droplets of ink that were generated and deflected by the methods that eventually became the same methods used in the cell sorter. My first involvement with cell sorting, I guess, was in the summer of 1963, which was before I'd actually published anything on the work that I was doing. But I got a letter from Mac Fulwiler, who was working at the University of California at Los Alamos, who had seen or talked to someone who had seen some of my work. I don't know now where he became aware of it. But he was working with Coulter counter, which was a device for analyzing cells in a flow system, and was feeling a need for some method of sorting the cells that he was analyzing in his setup. And as a result of some correspondence between the two of us in the summer of 1963, he came out and visited me and told me a bit about his work. And we had some conversations which were interesting to both of us, I think. I knew nothing at all about cells at the time. And he went back to Los Alamos with some borrowed equipment of mine and started work on what became the first cell sorter.
And where did your work on the inkjet printing go from there?
That was eventually became commercial device, and it's now used for mostly for product marking and unfortunately perhaps direct mail advertising. Most of the junk mail now is addressed with inkjet printers, and if you look on the bottom of Coke cans and beer cans that you buy in the supermarket, many of them will have an inkjet printed date code on the bottom. In fact, a lot of packaged materials that are date coded or marked on an assembly line are marked with inkjet printing now.
Okay, we'll get back to you and to Stanford. Now, Nick, where were you when the story began, and what were you doing?
I joined Stanford Genetics Department in 1962, and at that time we on some laboratory that is called Instrument Research Lab, IRL for short. And we were doing all kinds of things regarding space exploration and finding if there is life on Mars and all this kind of things. And we had quite a bit of monies from NASA. And when that work was a little bit slack, we decided, or at least the directors decided, that we should look into some kind of a cell machine that either will analyze cells, in the beginning mainly blood cells, but so I came into the project from the Instrumentation Research Lab under Elliot Leventhal, and we started looking at better ways of analyzing cells, similar to what Coulter was having an instrument, but maybe faster with better resolution. So that's how the whole thing started. And we ran into a lot of difficulties, as is expected, and mainly the difficulties is orifice problems. How do you make a small, very small orifice, so a few microns, not to clog or uniform, so that you can detect volumetrically the cells that they go through. We try to make orifices by our laboratory out of glass. We weren't successful in that. So the idea, I looked into I think the jewels, watch jewels, and evidently that's a technology that was all foreign to me, but sure enough, that was the idea that we should use one of those things to let the cells pass through. There were a tremendous amount of excitement and home-built type stuff at the time. As far as the separation is concerned, I guess some of the work that Fulwiler was doing was incorporated into this, and slowly we went into the volumetric first cell separation, but it wasn't very successful. And of course, Bill Bonner, another associate with the genetics department, he started to work on optical type of a machine, but there were tremendous amount of difficulties due to the fact that technology at that time was very, very primitive from today's standards. Computers were very, very non-existent, so to speak. Also, lasers were non-existent. So we have a tremendous amount of problems, and I don't know how much details you want to go into.
Well, before we go into maybe more details, we'd like to set the scene with Dick Stovel, just to find out when you came to Stanford and how you became involved with the project.
I joined the project in June of 1972, and I came here after a somewhat less than satisfying experience in aerospace industry. Not sure what I wanted to do after I was an aerospace engineer. Wasn't sure that I even want to stay in engineering. And I got a job with Len Herzenberg, whom I'd known from outside political activities, and I had a side interest in biology and knew he did something with biology, so I thought I might find something interesting to do here. And I started out as just as an operator, a machine operator, with the first cell sorters that were developed here. I considered myself at the time the world's second cell sorter operator, the first being Tim Merrill, who had already been working on the project for at least a couple of years before I got there. And gradually, as time went on, I gravitated towards the engineering aspects of the machine and became more an engineer than an operator. But my start was as an operator in June '72.
Going back to you now, Dick, we'll pick up the story. You did your work on the inkjet printing. Mac Fulwiler found out about it. You met with him. He was able to, I guess, apply or incorporate that work into the cell sorter that he was building. Now, where does Len Herzenberg come into the story, and how did this project come to Stanford?
Well, I had nothing to do with that. Len I believe, went to a technical meeting, which Mac gave a presentation of some kind. At least he heard about Mac's work in some way must have been 1964, early 1965. Perhaps it was 1965. And he arranged with Mac to get information. I know Mac sent him a complete set of drawings for the instrument that he had built at Los Alamos. And Len started a project with Leventhal's group, which Nick was a member then. And the other two members were, at that time, were Russ Hewlett and Bill Bonner. And I guess Tim Coburn was, Tim Coburn and Tim Merrill were also involved somewhere around that time, but I was not here. I'd gone shortly after the project started here, I left Stanford and gone to Varian Associates and worked on completely different things at Varian Associates. I was aware of the work here, and I came over and visited the group once or twice just because I was interested in what they were doing because of its relationship to what I'd done. But I had nothing to do with it until I actually joined the group in 1971. I came here from Varian and joined the group in January, February of 1971. And at that time, they had just gotten their first cell sorter operating well enough to do some biological work. They'd written the first paper. They were just firing up the first laser. The first work was done with mercury arc lamps.
Yeah, how different, was there any difference between the machine that Fulwiler was using at Los Alamos and the first prototype that was developed here, or were they more or less
Well,
copies?
the machine here was pretty much built from the ground up. They had sort of a false start in trying to copy Fulwiler's machine, and I don't really know the details of that, but I think Lynn decided that he was more interested in fluorescence detection than in volumetric measurements on cells, and the work went on, the goal changed to a fluorescence-activated cell sorter from the initial work of Fulwiler, who had done his work with volumetric sensing.
Primarily size of particles.
That's
that he
right,
was
particle
interested.
size.
And
So when I came, the project was completely divorced from Los Alamos. The work was all the work was done at Stanford by the Stanford group, and they were well underway to actually getting the system that was producing useful work.
I just wanted to clarify something with you, Nick. Now, was there any work in the exobiology program or concern about analyzing cells even before Len brought back the blueprints and started talking about the cell sorter, or did that start when
Actually, the exobiology work, it might have involved some technology, but it was entirely different. It was mainly activations of cells with some reagent, and then detections of activity by photoelectric type measurements. So actually the cell separation or analysis project had nothing to do really with the exobiology that was involving the detection of life on Mars, if that's what you were driving.
Yeah, I just wanted to make sure that to get the relationship between the two projects. Good.
Actually, we had some optical and some photoelectric detection equipment that would have applied, say, into the cell analysis and sorting, but I would say that it was the major problems in cell separation and detection. It was different from the exobiology experiments that we were performing at the time.
Let me pose a situation for you and have you react to this. My understanding was in part that Len had something very specific in mind that he wanted this machine to do to help him in his own research, and that he would come to you all, I guess, when you were developing the cell sorter and say, look, I want this machine to do this. Can you make it happen that way? Is that more or less right? Then you try to react to that and look at it from the engineering and technological perspective, and then say, yeah, we can do this, Len. No, we can't do that. I'm trying to get the dynamic between the engineers who were working at that time and the scientist or scientist in her chamber.
Actually, in the beginning, there were several groups that they were interested in some kind of a cell detection, either volumetric or optical fluorescence or whatever, and separation other than Len. But of course the impetus was Len, because actually he was part of the genetics department, and all of us were part of the genetics department. So we had to be directed to an activity that will provide an instrument that will help the cell analysis that Len was involved with.
And what
But-
would, I guess,
but
like,
we did have relations with the clinical labs, for instance, that they wanted to have some kind of an instrument for red cell analysis from the patients and so forth. Or Dr. Wiseman was doing some work in radiation of mice and so forth that wanted to have some cells analyzed and or separated. So there were several
yeah.
groups.
And I guess the name that has come up, I guess, what Henry Kaplan, who was a radiologist, was also interested and put up some money, developmental money.
I don't recall that. That's
Yeah.
probably true, because I left the project after, I guess, in '71 or something of that
It was
nature.
just shortly after I joined the group.
Yeah.
Yeah, and so by '71, was there already an operating prototype?
Yes,
I believe so.
yes.
The first paper, I think, what was, is it '69, am I remember correctly?
Well, I have it here. Len's first paper. I thought I had it here.
Well, we can find out
Yeah,
later.
anyway, there was
I
a
think it's-
paper published in Science by Len and Russ Hewitt, and not sure, there are probably other names on
Right.
the paper, but I know those two were on the paper, which was the first papers published by the group on fluorescence-activated cell sorting.
What were the sort of the engineering challenges that you had to deal with when working on this machine? You mentioned one, Nick, the orifices, or the opening the nozzles and getting, I guess, flow through in
Well,
and that's sort of fire high pressure, right, to get a, and,
to begin with,
Stop there for a second. We have to change tape. Excuse me.
okay.
Can anyone tell me what that roar is?
Rolling, hold for speed.
The question I was beginning to formulate was about the engineering challenges, the early engineering challenges that you faced when you started building this device. And Nick, you mentioned one about the opening of the orifice and being able to get
Correct.
the speed.
the
Mention a few
other
other.
major, as far as I'm concerned, it was the light source problems. The lasers didn't exist at the time, and we needed to have a strong light source to illuminate these fluorescent cells on the stream. We use xenon or mercury lamp, arc lamps to do this, but again, we had difficulties concentrating this particular light and into the size of the stream through lenses and mirrors and whatnot. So we even tried to manufacture mirrors, parabolic mirrors, in order to make the gathering light much easier and much stronger. And of course, mercury or xenon lamps, they have a wide spectrum, frequency spectrum. So in order to get down to the UV, which was most of cells are very active, by filtering and so forth, we had very little light left. Also, we wanted to try to pulse the lights so this way we would increase the intensity, and we had some successes there. But all in all, I think that the invention of the laser was the one that, or the affordability, I would say, of the UV laser was the one that saved the day. Others problems, we have, like I mentioned before, orifices problems. The other was stability and driving the column from the ultrasonic vibrator. So we had, because we could not put enough power onto the assembly to make the stream break in a specific area. So we had difficulties in that. We tried to devise all kinds of tuned circuits that will make it more powerful. But slowly, as I said, the technology improved and make it possible to have an instrument today that is useful in the medical field.
Dick, what about some of the problems that you work with, or are those similar
Well,
that you
Nick's
tackle?
touched on some of them. We were working with very tiny jets, of course. They were about, ones we started with, and still are, about 2 or 3 1,000ths of an inch in diameter. And the precision with which we wanted to locate the cell in the jet was considerably better than that. We wanted the cell to be in the center 5 or 10% of that cross-sectional area. So we spent a lot of time with the fluidics design, that is injecting the cells on the center line of the jet, being sure that they were in the center. It was important not only that the drops form accurately, but that the timing be precise between detecting it. There's a time correlation between the detection of the cell and its incorporation in the droplet. And that all related to where the stream broke off, and the things that Nick was talking about stimulating the stream so it would break off accurately. All the work that I'd done previously with inkjets involved fluids that had no suspended material, and it was something new to deal with objects that in some cases were nearly as large as the orifice, that is clumps and debris that were as large, or sometimes of course larger, which caused clogs in the orifice. We couldn't use the fine filters that I'd been used to using because they would have filtered out the material that we wanted to examine. So we had all the difficulties that were involved in getting the fluid system right, getting the cells to go down the center line of this 2 1,000ths of an inch diameter jet, get trapped in droplets at the right time. We had to deal with timing accuracies there of a few microseconds.
Maybe you can show that photograph, and that will help illustrate what you're talking about.
Yes, this was a picture that, actually this was taken by Bill Bonner, I think before I started with the group. And it does show a 50 micron, it's 2 1,000ths of an inch diameter jet breaking up into droplets at a rate of 40,000 per second.
And then the laser light would intersect.
The laser, this was taken with a light-emitting diode flashing at the droplet rate so as to freeze the droplets in the camera picture. And the laser, which doesn't show in this picture, would shine across the stream just below the nozzle exit here and illuminate cells as they cross that laser beam. The illumination time was only a few microseconds. So that was why we needed such an intense light source in order to be able to get enough photons out of that tiny cell in just a few microseconds to see a signal that we could analyze. The other big problem we had, or one of the others, was lasers. And these were new devices at the time, and they were entirely new to all of us. None of us had worked with lasers before, and they took a lot of getting used to and caused lots of problems. In fact, they still do. But at that time, everything was new, and we spent a lot of our time trying to figure out what was happening with the lasers and why they didn't work the way we expected them to.
Can I interject a little bit about the nozzle technology? The question of how to make a good nozzle is still a very active one. I've had discussions with Beck & Dickinson engineers as late as this week about that very issue. And it's interesting, a good fraction of the nozzles these days are made with watch jewels, tiny little jewels with orifices in there made for watch purposes, but were adapted and used in cell sorting technology. That picture that was shown was actually a drawn glass nozzle, which we used for were in use when I started and for several years thereafter, but had obvious disadvantages of breaking. But I was just talking with Nick recently about the origin of the watch jewel, and found out that he found out about watch jewels way back in the early part of the thing by going downtown to some jeweler. I don't know how you got that idea, Nick, but they went out of fashion around here and came back in, and the commercial machines, and now there's a lot of them in the world, but they're now also using ceramic nozzles as another alternative.
Before moving off the fluidics, because we might want to get onto the electronics and the computers, maybe you can bring up the story, since you said that when you came and started work, fluidics was still a problem area or a developmental area. Maybe you can speak about that a
Well,
little bit.
in the prototype machine that was in use when I started, the samples themselves, the cell suspensions, were put into very large tubes that were, I don't know, I think they were about 20 milliliters in volume. And by current standards, that's a very big volume. And people hadn't learned to think small in terms of the cell size objects. And we had at that, when I started working, there was a engineer who was kind of a classic crazy inventor type named Ken Shoulders, who worked here just part-time, but was very clever at miniaturizing things and made quite a few flow components for the prototype that was coming together in 1972 and later became our primary home-built machine called Wizard. And he he based this technology on these molded elastomer parts that he built special molds, and he was challenging our machinists to make very fine mold parts. And I can show you some of the things that he did. He molded lots of little parts like of this magnitude, which handled the cells internally. These are valves and connectors type things. And he made various versions of these, and he did this by having the machinists manufacture these molds. I mean, I'm sure these pins are almost too small to see. Some of the internal workings of these molds are so small. They're almost too hard to see. And he made a lot of components, very intricate detail. Here's some other molds over here that we use later on in these boxes. And he came up with a set of fluidics that, for our prototype machine that involved all these things. And some of that technology persisted around for quite some time. And some of it fell into disuse because only he could manufacture these little tiny fabricated parts. And the Becton Dickinson, when they made their first machine, designed their own set of valving for the fluidics, which I have, I think this is actually the one that was on the original Fax 1 that was delivered here. And it did not use this molded RTD, I call it blue goop technology, but it used a different kind of sliding valve, a very finely engineered kind of thing, but it actually didn't work out too well. For one reason, in those days, our primary means of sterilizing the machine was by autoclaving, and this part was made out of acrylic, and it was not autoclavable. And I had other problems. They eventually tried to make the thing out of ceramics, which was autoclavable, machinable ceramics, and they had a version of that didn't work too well either. And they ended up, when their early machines, coming to us and saying, well, how are you doing it now? And by that time, we had gone on to some more of these things. Ken Shoulders had left, and I had started making these things myself and used them for pinch valves instead of connectors. And so I was making these little tiny blue goop valves, and Becton Dickinson decided that they would try to do that rather than engineering their own thing. And they built, for a long time, in the early machines, they built these blue goop valves, which I manufactured for them in the early days, and they picked up on how to do it. And gradually, that technology died off because the good quality silicone rubber tubing came in that we could use directly as pinch valves without having to make connections. But Ken Shoulders was certainly a colorful guy, and for other reasons in the early days.
You mentioned the silicon, sort of conjures up the image of Silicon Valley. You're here.
Yeah.
So I take it these things are...
Silicone rubber, it's the same element in a different form.
Nick and Dick Sweet, maybe you can address this issue. Now, tell us about the workplace where you were working at. I mean, we hear stories of this all was happening in the basement of whatever building you were in at the time, and it conjures up, rooms and tables with bits and pieces everywhere, and machines in various stages of development. Well, give us a truer picture of what what the laboratory and what your
Well,
workspace looked like?
At the time, the laboratory was very active. We had very few people, but all of them that were very busy in one way or another. We were involved in mass spectrometry, we were involved in cell separator, optics, trying to make lenses, trying to make valves, trying to make everything that we could find, nozzles, glass blowing, and so forth and so on. At the same time, Kaminsky from IBM, he delivered also a machine to us, that some of his early adventures in cell separator or cell identifying and so forth and so on. So it was a very hectic type, or not hectic, but enjoyable type operation, and we tried to have some breakthrough in all these adventures, and we managed to do with time pretty well, I think.
So you were looking at for example, the Kaminsky's machine and taking it apart, see how that worked, and looking at what Fulwiler was doing, and then trying to come up with ideas that might be useful from those machines to incorporate into what you were doing. Is that?
Kaminsky's machine had pretty much been abandoned by that time. By the time I came, it still existed down the hall, but nobody was looking at it anymore.
Well,
in terms of the actual cell sorting method that we were using.
Because that was primarily for dead cells, right? And you were interested in live cells?
It was different technology, which never did really work out. But Kaminsky was also active in flow analysis. And there, he continued to make contributions, and I think that some of the things that we did certainly profited from his work and his continuing work.
Talk a little bit about the early involvement with Becht and Dickinson, because obviously they had a group of engineers working there, and you were working here. What kinds of interactions were with these two groups, and how did the information flow back and forth?
Well, we had an operating sorter by the time Becht and Dickinson arranged the contract to build two systems, and their charter really was to not to copy what we'd done, but to produce an instrument that was a manufacturable device that would duplicate the performance that we had. So they had to do a lot of things differently than we did, but we worked quite closely in terms of giving them all the information that we had gotten by that time and how to do things. We operated very informally. We didn't have conventional engineering drawings. Most of the things were done by engineer sketches, engineering notebooks, and just conferences and
Show us
things
some
over
of
in the
these
lab.
examples. Do you have any ideas or examples of the kind of notebooks that you were keeping at the time? You can show
Well, we
the
have
drawing.
notebooks here. I don't know. They're mostly data. The trouble is that I don't think we have. Do we have anything showing drawings or sketches of things that we made at that time? I don't. Other than schematics, we really don't. I think most of the mechanical engineering work that they did was done by making their own drawings, probably from the actual parts. They would borrow parts from us, or they would come over and look at how we did things. But the actual machine that they built had nothing that was directly copied from us in terms of the actual mechanical arrangement.
If I understand it correctly, Fulwiler had a patent on his machine. Now, then Stanford got a patent for their
We got a
machine?
patent on the improvements, some of the improvements that we made. I think it's fair to say that the pioneering patent was Max, and that was licensed to Becht and Dickinson by the Atomic Energy Commission, which was the owner of the patent.
How would you characterize maybe the lifestyle of the laboratories? I mean, you said a little bit hectic, but you probably meant busy. But And I guess from what you were saying, Dick, is this fairly informal exchanges of ideas. Obviously,
That's right.
probably you didn't know exactly what you wanted to come up with, so you couldn't set an end date on when the project had to be completed because
No.
it was an
We
evolving.
we had goals and directions from Len, of course, who knew what was useful to him and what he wanted, and we did our best to meet those. But often it was just a question of doing the best we could without knowing how close we would come to what he needed. We were always looking for more sensitivity, and that meant getting more light on the cells and increasing the sensitivity of the electronic processing. I don't think anything's been said about the electronics, so that was a good part of the work, was designing. Initially, we bought modules from companies that made amplifiers and peak detectors and gating circuits and so on, but we finally ended up by designing all of that circuitry ourselves, most of it, I should say. That actually grew into a fair part of the project, particularly when we got into computers and data processing.
But in the early prototypes, you had the electronics. Did you have an early computer?
We had,
we had
a Link.
we had a Link computer, which is the initial attempt on computers, and then DEC picked it up and make the PDP, whatever, 8 and so forth. And we tried, but of course it was very difficult to couple the computer onto the instrument at that time because there was no software and so forth. So most of the stuff that we did was essentially handmade electronics, amplifiers, delays, and so forth, because there was no such thing at the time. And so that, in a way, was a hindrance in improving an instrument, because we had to build the electronics, we had to build the instrument, and we weren't that many of us. I mean, we were, for instance, at the time, we were Bill Bonner, myself, and maybe a technician, and one programmer that was doing all this work. And Bill Bonner was on on a cell separator based on photosensitive devices. I was in the volumetric measurement. So we had a machine shop that we were doing the work, but most of the drawings have to be done by one person, mechanical, electrical, electronic, or whatever you're going to have. So it was a very busy time for us. As far as goals are concerned, we try to do our best.
Rolling, hold for Steve. Steve?
Maybe to wrap this first session up, I'd like to ask you, was there any time in the project that either you or the scientists involved threw up their hands to say, no, we can't do it, it's not doing what we want it to, let's forget it? Or to some extent, not necessarily in those words, I mean, but was there any kind of point of despair when things weren't going right, where it seems that maybe there wasn't any future for a cell sorter?
Well, to begin with, we started with a volumetric analysis, and somehow or another, either that thing was not going very well, or it was taking too long to develop, or there was no interest in whichever of the three, or funds were drying out, whatever the case might be also, it was dropped. But there was a tremendous amount of learning that was involved in this, and that's what a research institution is all about. And I think initially, even the optical cell sorter, for instance, it might not have turned out to be the way it did. It might have been dropped also, but luckily Len was able to acquire the funds to continue with the project.
I think when I joined the group, there was a great deal of excitement about the potential for this, because they just really started achieving meaningful successes. so it was a really nice time to get involved in it. And I think probably the doubts and reservations were whether it was going to have any commercial potential. This searching for somebody that would be willing to try building some and selling them was a bit frustrating, although I wasn't involved in that. And I think there was some skepticism on people, both in our group and people we talked to, about whether this was worth pursuing commercially, even though it was biologically and scientifically very exciting.
I might interject it when from my perspective as an operator, when I was working, when the BD was coming out with its machine, there's certainly internally a lot of optimism and enthusiasm about the machine that we were operating, but there was also a lot of doubt in our minds as operators that BD could come up with a machine that, based on what we were doing, without a lot of years of development, that would be usable by the public, because there were so many things you had to nurse in operating our prototype. And I think in retrospect, we probably did better than we thought they were going to do, and machines were used by people. And I think a lot of those early machines sat idle because people didn't know how to use them or couldn't use them. But eventually, the technology came along, and it was. I think there was. I didn't have any sense that there was any vision amongst our group that this would be anything like the commercial success it has been, except for possibly with Landon.
Yeah. Well, that's a great lead-in, because I wanted to ask both you, Nick, and Dick Sweet, because you were there so early, how you view that whole development of cell sorting, flow cytometry, and the facts now that we see what the fourth, fifth generations coming in. What's your thoughts on that system?
I think it's something that, say, we started here at Stanford, and it turned out to be very successful. Although I said earlier that we had very, very much difficulties, and we hesitated. We didn't know that this is going to go anywhere.
Is it something you tell your grandchildren about?
No, I suppose so.
Sure. I think that the part of the project that we were in is the most fun part, the most enjoyable part of the development. The real early discoveries and getting things to work, and then the actual producing them and making money with them and doing things right rather than the chewing gum. And the way we did them there's a lot of value in returning that, but for me, the excitement and fun is the early stages of the project.
And I guess maybe before we end, there's another question now. You've all had other lives too, and maybe briefly you can say what other projects you have worked on subsequently. I don't know, maybe as exciting or less exciting, but just to put your sort of careers in perspective here. Maybe we can start with you.
Well, my background was in electronics. I'd done that ever since I got out of college, and that was what I was doing at Stanford at the time I started the project on inkjet printing, which was a big diversion for me. And that was probably the most interesting thing that I had done up to that time, was the inkjet printing. And of course, I've been involved in things related to that ever since.
You, Nick? What have you gone after the cell sorter? What?
Well, after the cell sorter, I started getting involved with mass spectrometry, and I'm an electrical engineer by trade. However, I'm thankful that I can work on any type of mechanical or vacuum systems and so forth. So we were heavily involved into mass spectrometry, data collection, including computers and so forth and so on. And later on, when that project finished, right now I'm with a Summix computer project, which was initially with the genetics department, now is with the Department of Medicine, and we heavily invested into networking and so forth, and advancement of electronic mails and things of that nature.
And you, Dick?
Well, I think that the working on the machine in those early stages, in a lot of ways, it was the good old days. It was a fun place to work. Dick was a had a relaxed managerial style, and having fun was an important part of it. But we had plenty of success to feel good about, and that success continued as the commercial machines developed. And so I think we've had interesting projects since, adding pieces on to the sorters, but there's nothing like being in on the ground floor and seeing it take off.
It's great. Thank you very much.
Okay, I'd like to do just a brief little bit of reacts here,
Okay.
okay? So maybe just stay on, what we're going to do is just shoot you a little bit, listening to Ray, okay?
Listening
This is
to-
for editing
okay.
purposes only. And you can either talk, why don't you talk to him for a second?
Okay. Well, I found it very interesting to come here and seeing all the pieces come together, and that's one of the things that sort of interests me.
Rolling, hold for speed. Speed.
It is now very late in the afternoon, and we're talking with our second group of engineers who have worked with both.
Here we go. Is that you hear him now? Speak into the microphone.
1, 2, 3, 4. Sorry.
Try again? Go ahead. Still rolling.
It's very late in the afternoon now. We're talking with our second group of engineers who have worked both with the early fax machines and now with the next generation of instrumentation. From right to left, it's Wayne Moore, David Parks, and Tom Nozaki. And maybe we can start with you, Wayne, saying when you came to Stanford and when you started working with the cell sorter.
I believe I came to Stanford originally in 1971, and I was working what then was upstairs on the third floor in the Herzenberg's actual laboratory. But I had had experience with computers both in my last year of high school and as an undergraduate. And so when the first PDP-11 came onto the project, I sort of took it over and then moved downstairs into the basement where all the engineers were. And that was, what do we figure, 73. I've been there since then.
So computer programming is
Yes,
your
I do.
pro-
I've been doing software the whole time.
David?
Yeah, well, I got into the group in the fall of 1975, although I had been a graduate student at Stanford in nuclear physics considerably earlier, and remember minor conversations with Len Herzenberg, who I knew from other things going on at the time, and maybe a skeptical question about, are we wasting our time trying to get into all this kind of thing. But I wasn't really involved in it until
in nuclear work.
No,
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 or
the
in
project that he was on, and he was trying to deal with this engineering thing, which he, as a biologist, didn't understand entirely, and wasn't sure that he was in a good place for really having it carried through. But as we heard earlier, that did come through quite nicely. And in 1975, I joined the laboratory as a research postdoctoral fellow. And then after several years as a fellow, switched over to being a regular appointment research associate in the Department of Genetics. And I've worked largely with one foot in the biological side and the other in the instrumentation, trying to deal with the physics and optics and things like that, and also really do some biology. My interests are largely really biological, but my background puts me in a better place to work with the instrumentation. And then when it came around to making the capabilities more available to other people besides the one laboratory in what is developed now into the shared facts facility, I was involved in coordinating that, and has become probably half of my total work now, is just directing that facility.
It's the one we saw this morning. You gave us a tour.
Right.
Tom, same question to you.
Yeah, I started in 1974. This was my first job out of electrical engineering school. And previously I was working at Stanford for the ACME project, which was an NIH-funded project, which was one of the first time-sharing systems. I was in the engineering group, which designed and built interfaces for different scientific laboratories and medical laboratories throughout the university. And so after I graduated from engineering school, this was my first job.
Now, why? So I take it your task was then to develop interfaces with this instrument, or?
Yeah, they hired me because they didn't have any computers with the cell sorters per se. They had this Link 8. That was a kind of very old computer, and when I started there, they just had purchased a GT44 system, which was a PDP-11, Digital Equipment Corporation type computer. And they wanted me to interface the cell sorter. At that time, they just had the new First Vax 1 cell sorter, which was a manufactured cell sorter by Becton Dickinson, which was from the prototype that was developed at Stanford. And so I was hired to interface both cell sorters to the computer.
And I guess the program then, mentioning PDP-10, that's where you came in, right? Wayne, and
yeah.
developing the program. And
On that early, the 1110,
yeah,
which is the CPU and what he was talking about.
Right. What was the challenges and the tasks there, the programming, the problems that you had to deal with?
Well, the PDP-11 is a very limited machine in some ways. It has a very limited address space. There was virtually no operating system software for it at that time. When we got it, it had a paper tape operating system. We actually first installed it up at the AI laboratory, and we used the Artificial Intelligence Laboratory. This was out when it was out on Arastadero Road in the big round building. And we actually used the computer there to bootstrap it up into something that we could use. And their motivation for it actually is kind of funny. They wanted to use it as a memory controller, and so while it was sitting up there, they developed a space war program on it as their way of validating if the thing worked and evaluating its performance and how it was going to do for them. And I used the tools that they developed and some additional tools to get the first. It it was a home-built operating system. It wasn't the first operating system for PDP-11, but it was a simple home-built operating system that we used that was booted off of floppy disks, which I think were also, we had very early floppy disks, because when that became standard, then ours didn't work anymore, but we had the very, very early use of floppy disks, and that was the first round of software, which I guess we're now at the end of the third. So.
Third generation
Yeah.
of software. David, you watched the evolution of these machines and permutations of them as the science changed, and I suppose as you described yourself as having one foot in the biology and the other in the physics world. Talk a little bit about that dynamic, what was happening in biology, the different kinds of questions that they wanted to get at, and then how you go about finding instruments to give them answers.
Yeah, well, one thing I think should continue from the other interviews is the question of what was driving the particular projects here. And that is somewhat different than other flow cytometry development projects. The original factors of the immunofluorescence measurements that Len was interested in because of the kind of immunological biology that his lab has interested in, as opposed to, say, a lot of other people who are interested in DNA measurements, which are fairly different in a number of ways and have important implications for what you do when you're building the instruments and the software and everything else. There was the emphasis on working with live cells and being able to actually sort the cells. There's been, and always has been, much more work in cell analysis, just making measurements on cells, which may as well be dead because you're not even going to consider recovering them. And, in fact, done a lot of sterile sorting, where the cells are kept in a state where they can be put into long-term culture again and grown up and that sort of thing. And a lot of work for developing rare cell isolation techniques. And then the other dimension of that has been the development of the fluorescence techniques, particularly multiple immunofluorescence, where you can measure several different cell surface molecules or sometimes other kinds of cell properties by separate fluorescence measurements in a single pass. So each cell is measured on a number of dimensions. And the use of monoclonal antibodies was very important in that, making it possible to have a number of different reagents, each coupled to a different dye, and then measure them all independently. And then most recently, that has started to flow off into adding physiological probes and other things like that, along with multiple immunofluorescence, and get a potentially very complex sets of data that you're dealing with. So that's been, I see, sort of the flow of emphases around here.
I'll stop here for the talk.
Good.
Hold for Steve. Steve? Hey, Ray.
We have seen in our discussion that neither science nor technology are static processes, but there are dynamic processes in themselves, and they interact and affect one another. And maybe, David, you could talk a little bit more about the incremental developments that need to be made to these machines to keep them from becoming static entities.
Okay. Yeah, well, so relating to the emphases that I mentioned a while ago, there have been a number of developments in the sort of engineering direction, say, for myself, I would say other than the computer and software side of it, that I could go into a little bit. One of the earliest ones that I was involved in is called fluorescence compensation, where you may be exciting more than one dye, at least two dyes, with a single laser beam. And the fluorescence emission from those dyes isn't entirely distinct. And therefore, you can't put a detector out and measure a single value that relates only to one of those dyes. But we developed techniques to correct for that, in a sense, and be able to get effectively single dye signals out after doing the measurement. So that made it possible to get 2 and sometimes 3 different measurements, fluorescence measurements, off of a single laser beam and still keep them distinct. So that was one early important thing that's in totally routine unit use all over the place now. And that was expanded out by getting a second laser beam into the systems so that we were doing 3 and sometimes 4 color immunofluorescence. And it's important there, the immunofluorescence often has very low signal levels compared to like DNA staining and things like that. So that's put a lot of pressure on the sensitivity and quality of the measurements themselves. And that, another thing that came up fairly soon was the question of cell cloning. As long as we were sorting cells thing, in a lot of cases, since we were doing sterile sorting and wanting to culture the cells afterwards and isolate particular kinds of cells and rare cells and things, you want to grow up clones of those cells. And so it seemed like a good idea. Well, as long as you identified a candidate cell as a likely one that you want to grow up, put it individually in a well and grow it up. And that was one of the things that we designed and built accessory electronics, basically, to carry out that function, plus the mechanical objects to do it. And in some cases had to go ahead and build more copies of those to distribute them to other people, which in particular Tom has been instrumental in doing. We developed a sampler from 96 well tray, making it possible to do cell staining in 96 well small trays, use small amounts of reagents, small numbers of cells, handle as a whole set in one, which improves the uniformity of handling, of course, too. And then go in a little microprocessor controlled device and grab a sample out of each well and put it into the analysis work. The 4-decade log amps were an important factor also. And this is another factor where the immunofluorescence and that kind of emphasis is important. DNA staining, you have essentially a 2-fold range between for typical kinds of proliferating cells, and you just want to make good, accurate measurements of those. Immunofluorescence often has 1,000-fold range between unstained cells and brightly labeled cells, and you want to see everything in between at once. And so the logarithmic amplifiers, which had been proposed and used somewhat in a number of other places, but with limited success. In particular, Dick Sweet took that on, and with changes in what was available in the electronics to work with, and some good and somewhat clever ways of using that, came up with effectively 4-decade capable amplifiers, and which, again, Tom supplied to many people for a long time before the commercial sources caught on and started making those sort of things available. I guess the other thing, a number of factors, including isolation of fetal cells from maternal blood and isolation of mutants. We've pushed hard on selection of rare cells and being able to identify and then sort out 1 cell in a million in some sample and catch it and grow it up. So that's kind of a rundown on some of the...
This is a pun intended, Tom, but could you amplify a little bit about some of the work that David had mentioned, and once these ideas or modifications are developed here, and how are these ideas spread or instruments spread to other areas, and talk about your own work in that regard.
It's mainly the Herzenbergs. They have many postdocs that leave the laboratory, and they want to continue on doing the same kind of research. And they don't have the same kinds of tools that we have. They're not commercially available. So they asked me if I could do something for them where I can go and build the log amps in this case, so that they can use it in their own laboratories. And like David said, that that it wasn't commercially available. Beckman and Dickinson, at one point, they came to us to go and try and duplicate this system, but somewhere in the duplication they didn't follow exactly what we did, so they weren't as good. And so even though they produced the log amps, they weren't as good as ours, and they still came back to me for manufacturing the same kinds that we have. And so it's mainly all the conferences where people go, many immunology conferences where they see this data published, this log data, and they want to know how to do that. And it's sort of word of mouth, and they come back to me, and I produce them for the people that need them.
This morning, Wayne, when we were on our tour, we were looking at the screen with the new sort of software that's being developed for the cell analyzer.
Mm-hmm.
Could you talk a little bit about the sort of the current challenges that are facing you, the kind of software that you're trying to develop, and where you see the future going?
Well, the current change is sort of associated with a major change in the way computing is done, which is workstations. Historically, we've been using character cell terminals, essentially where you have a rectangular grid and you can put any character in, but you're constrained to that grid. And the new workstation is you have a very high-resolution screen, and you can essentially hit any point on that screen, and it's an entirely different style of computing. We're trying to take advantage of that to give you everything in an integrated environment. Typically, we had dot plots sitting here and the terminal sitting here and different things, trace displays over there, and we're trying to put that all together on the screen. We're trying to extend the analytic package, taking use of that flexibility. We want to add multiple levels of gating, for example, to the data. Currently, you have the sample, then you have one kind of analysis and one level of being able to display it. And we'd like to be able to put multiple levels of analysis, so you can say first, okay, these are the lymphocytes, and then these are some other type of the lymphocytes, some subset of those, and that naturally maps into a tree kind of description on the screen, and you can actually put up a tree then and have somebody point and say, okay, I want this analysis applied to this sample. We're also have a project to develop database technology, object generation in databases with this to aid people in designing protocols, the sort of the plan, the recipe for an experiment, if you will. We currently have a system where, you want me to explain the background of this a bit? It's when people first started out in the early versions of the floppy disk system we were talking about, people would come in and they would run their samples, and there were only 2 parameters, so you could collect a 1 or 2 parameter histogram and store it, and that was no problem. With the development of the multiple immunofluorescence, where there's many parameters, it wasn't feasible to store histograms anymore, so we needed to take list mode data, which was much bulkier and needed other ways of processing it. And after operating for a while, we had stacks of floppy disks that were useless because nobody had any idea what was on them after the people left. So we developed a first round of protocol editor, which essentially let people specify what they were going to do before they went into the room in a simple way, so that when they actually went into the room, they would just point at a location in the protocol saying, okay, this is what I'm doing now. And so all that annotation information could be put into the computer with the data, and then people get their plots out saying this is anti-IgM on bowel spleen cells, instead of not saying this is sample 1 and P1 and F1 or that kind of thing. But that system doesn't really understand the protocol. It's very mechanical. It's just saying this is here, this is here, this is here. And we're now trying to develop systems where you would actually, to some extent, describe what you were trying to do with the experiment, and then the system could find in the databases reagents that would be appropriate for that kind of experiment, could warn you if you'd done something that was obviously not going to work, that you know that this is going to react with that, so it won't do what you think it will do. Let's see, what else? And also, I guess, standardization and calibration has been a big issue with us. We've developed methods based on fluorescent bead measurements for automatically calibrating the machine and now compensating the machine. And we're in the process now of kind of wiring that into the system in a central way. It's kind of been hanging around on the edges for a while, but it's now going to become a very central part of the system.
Do you guys talk to each other every day? I mean, what's the work like? Do you go off and work on your own problems for a while, or how much interface there is or overlap in what you three doing, and how do you deal with that?
Well, you know, the group has significant overlap, and in particular, since I like to be able to leave for a while at times, I make a point of seeing that I don't do anything that someone else can't do. Occasionally, Wayne and Tom both do some things that no one else can do, and we're stuck if they're not around.
But I don't like to travel.
Yeah.
Go to the Andes Mountains and photograph insects, right?
Right. So we do have a lot of, I would say, sort of overlapping among the members of the group, so that we do have to work together and interact and choose who's doing which particular thing, although there are certain things that really only one person. It's a small engineering group, and also partly because we're supporting this shared cell sorter facility, which takes a lot of our time and energy just to keep it going and to keep new things, get things implemented in it.
Do you train other people to operate? Is that part of your...
That's part of it too, so it takes a lot of my time and a lot of everybody else's time. Everybody here is roughly half time working for the facility itself, just basically serving users, although part of that is making the new developments available within the fax facility so we can, in a sense, sort of test them out on a wider array of users than we would get if we were, this has emphasized the Herzenberg laboratory and stuff out of the historical thing, but in fact, we deal with, I don't know, people from over the last couple of years, say 50 or 60 different laboratories in the facility. So that provides a very different source of inspiration and problems.
Well, it must be exciting in some ways, sort of being at the sort of the edge of science in some ways, and I'd like your reactions. Today's New York Times, there was a story about new developments in AIDS, and part of the work was done here, and obviously a lot of it had to do with the cell sorter and what you all do. Just wanted to get your reactions to things like that. How does it feel picking up the paper and seeing your work? And any thoughts? I mean, or is that are you just a routine part of your... I mean, it's hard to sort of get the perspective. But,
yeah, well, no, it's clear that the kind of measurements that are involved here are kind of fundamental to important processes and potentially medical applications on a day-to-day basis, except for the AIDS work that we're associated with in the Herzenberg lab. We don't have that much interaction with direct clinical kind of things that tend to make it right out into the papers. It's more basic research.
scientific papers rather than commercial papers.
Yeah, although it makes it out there. Some of the stuff the muscle cell people are getting some press lately too, for techniques that I helped them develop and things like that. And so that's an interesting kind of thing. I don't know, maybe I'd say that one of the important factors here has been that there are a lot of different researchers that we do have contact with. back when I started in 75 and 76, go to meetings or something, and people would say, well, and that's all well and good for you to be able to make those fancy kind of measurements, but the rest of us can't reproduce them. We can't build on them. And it's cute, but it's not really very interesting as a general technique, and this won't ever be something that people do routinely. And at this point, it has come around to where, especially the analytical measurements, are very routine in a lot of places, although I think our facility still has more capability of really supplying routine four-color immunofluorescence, things like that, and really reliably getting good data of that sort, doing sorting, sterile sorting, things like that on a totally routine basis, which in a lot of competent research laboratories, they still shy away from doing cell sorting, or at least sterile sorting and this sort of thing. So that's been an important factor in making these applications of capabilities available for people with different kinds of applications. So people, particularly right now from all over Stanford, can come in, get both consulting help in applying the technology to the kind of things they're really interested in doing, and then going ahead and actually getting reliable quality measurements on each of them. So that's been an important capability here.
I'll give each of you sort of a chance last to hear some thoughts on both either your observations on the cell sorter or what you're doing now, or how you see your future career developing in this
Well, like
field.
I said, this was my first job out of engineering school. I've never worked anywhere else. I don't think I've ever interviewed anywhere either. But, right now, we're working on a new advanced pattern sorter, which is really exciting. And I don't know, that's about all I know is the cell sorters. And it's real interesting work, and I think I'll continue on doing this and stay with a group. It's a group that's been around ever since I started. That's one thing about this group. We've kept together, and I haven't seen anyone leave, and we've stayed together for, like me, 16 years together here, and that's a long time for someone, have a group and don't have a turnover of moving on or anything like that.
Do you share those sentiments?
It's a little different for me. I mean, I've been here for 17 years as well, but it's been 5 different jobs. I mean, we started out on PDP-11s. There were 2 versions of software in PDP-11 in a small mini computer. Then for a while we got diverted into these little pipetting robots, the auto sampler for the 96-well trays. We also had an auto deposition system where if you can sort single cell for cloning, you can sort it into a slide so that you could come back and look at it with a microscope and compare the flow information with microscopy. Now that technology hasn't actually been very well, that hasn't really gone that far, but we did develop, the technique to do that, to move the slide, and we had an automatic pipetting machine. And so we were using these little tiny microprocessors to do that, and then we went.
Stop right there and change space. We're ready to
Yeah, well.
Okay, then after that, I guess we got the 780, which was the first of the so-called super mini computers. That was sort of a major process to get that all in and get the room built, because it needed a cooled room and everything like that. But then we went to, previously we had basically had single user computers, but this was a computer that was actually running a time-sharing system, and so we had the same computer serving multiple instruments, and developed a much higher level of support that was available, so we had much more processing power. Nowadays, well, then I guess there was the pattern sorter project, the whole design of that, to try to move the sorting decisions away from a purely hardware implementation to a software implementation that would be more flexible and capable of reproducing some of the more advanced analytic techniques that we were developing. Nowadays, I'd say that the computer technology has largely caught up with us. I mean, in the early days, as I said before, we actually had to write the operating system, and we're certainly not in that position anymore. And almost all of the things we need that aren't specifically fax related we can buy or have given to us preferably. And so I'm moving more into data analysis, looking at new techniques for visualizing and analyzing the data and being able to focus more on that aspect of things than actually the nitty-gritty of software development. So the job has changed. I mean, it's the same place, but it's been many different jobs.
And different challenges.
And one thing that I've always really appreciated out of it is a lot of autonomy. I mean, we do obviously have to work very closely together, but on the other hand, I can cut off my corner of the project and drag it back to the library with me, and come back in a month or whenever I've got something and go over it with people. And for me, that's a very desirable situation, a very productive one. I think it's worked out well for all of us.
I'll give you the last word, David.
Yeah, it's been interesting and it's a full way to work, although I have to say that when my wife wants to give me trouble, she says, well, explain to me your job description. Because I was gone from being a postdoctoral fellow where I had specific kind of projects, some biological and some more instrumentation, to where now I'm partly directing a shared facility and serving a lot of different people, partly trying to coordinate this, I don't know, what do you say, small but still sort of sprawling group, and partly trying to still do some biological research done. And I'm never satisfied that I'm doing any of those adequately, but I keep plugging away at each of them and refuse to just simply drop things and cut them back to things I can do adequately. So I guess I sort of deserve what I get in that.
Well, thank you all. I think you've shed a lot of interesting light on this project, and good luck to you all in the future.
Thank you. Thank you.
Okay, let's stop tape for a second here.
We're rolling, stand by. And we have speed.
Okay, we are looking at accumulation of pictures that we've taken over the years of the people that worked in our laboratory. And as mentioned before, there was one person that has worked on the original cell sorter and designing it, and that's Bill Bonner, this picture up here. And he was one of the inventors of the cell sorter and shares the patent. And then picture over here is Mac Fuweiler, who we mentioned was working at Los Alamos and doing the developments on the cell sorter at the same time.
Okay, put it in pause just a second. Is this,
Stand by. Speak.
And in this picture, we see Lee Herzenberg in the earlier days. And down here, we see a picture of Len Herzenberg.
this one? That's Len and Mac Fuller. Tom, will you identify it?
Okay, in this picture, we see, on this side, it's Len Herzenberg, and in the middle, we see Mac Fuweiler.
And on the far right, is that me? Or not identified?
No, I don't know.
Okay, good. We'll finish with that card.
Stand by. Speak.
And here's another set of pictures that we have. And in this picture down here is Ken Shoulders, who Dick Stovel mentioned earlier. And up here, we see a picture of Tim Merrill, who was one of the original cell sorter operators on the prototype that we made at Stanford. And over here, we see a picture of Bernie Shores, who was owned the company of the people that came to Stanford and manufactured the first cell sorter, so it was commercially available.
Room tone in the library. Room tone.